Hello everyone. Today we are going to discuss protein synthesis. So last Friday I talked about cancer and how a mutation or several mutations in DNA can actually lead to faulty proteins that are supposed to regulate the cell cycle but are unable to do that, which leads to uncontrolled cell growth and division. So I want to go over the specifics today of how DNA actually codes for proteins and what that process looks like. All right, so before we start that, let's just go over why proteins are so important and why I consider them to be the most important macromolecule that we learn in this class. Most of the work, if not all of the work, is done by the cells using proteins, and proteins are the result. They're actually doing all of the work. We know that enzymes are proteins. We know that proteins facilitate transport of substances into and out of the cell. We know that proteins are involved in cell-to-cell recognition. Now, carbohydrates are going to play a big role in that too, but it's a protein that a carbohydrate is actually attached to on things like your blood cells that allow it to even play a role in recognition at all. So proteins are really, really important in cell-to-cell recognition. Antibodies. Antibodies are proteins made by certain white blood cells that are able to disable viruses and bacteria. So what an antibody will do to a virus is it will surround it, making it not able to dock onto a receptor or attach to a receptor on the host cells, and then the virus can never gain entry into that cell. Structural proteins are going to make up connective tissues such as ligaments and tendons. So proteins are involved in structure. And then some of your hormones. So when we think of hormones, we usually think testosterone and estrogen, but a specific hormone that I want to mention here would be insulin. Insulin helps regulate blood glucose levels, and insulin is a protein. So some of your hormones, and a hormone is just a chemical messenger that travels through the blood. And so some of the hormones, some of the chemical messages that travel through your blood are considered proteins. And then enzymes. And my screen is kind of cut off at the bottom there. Let me move that. There we go. We did an entire day on enzymes, and enzymes are going to speed up the rate of chemical reactions, remember, by lowering activation energy without being changed themselves in the process. So they're very, very important. This is just showing you a slide of the Krebs cycle, also known as the citric acid cycle. It's just showing you all of these different steps that take place in the citric acid cycle are achieved with the help of an enzyme. That's all I'm trying to get you to see here. Enzymes usually end in the word "ase" and are necessary components of every form of chemical change in the body, such as the steps in the citric acid cycle. And so I think I drove home the point on enzymes pretty well whenever we went through it, so you guys already know this. Okay, and speaking of insulin, remember, we talked about the folding of a protein being very, very important. And so just to recap on what proteins are made of, remember, there's 20 amino acids. And so how do you recognize an amino acid? It has C, H, O, N, and sometimes S. But once you get your entire chain of amino acids, and a chain of amino acids are called polypeptides. Chains of amino acids are called polypeptides. It's just amino acids that are covalently bonded together. You are going to get one of the amino acids that has sulfur. So that's why we say if you have a protein, you're going to have C, H, O, N, and S. C is carbon, H is hydrogen, oxygen, nitrogen, sulfur. And so that's what you're going to see. So insulin is our example here. This is what the chain of amino acids is going to look like. And like I had said, it's a hormone. Insulin is a hormone that regulates blood sugar. It's made up of 2 polypeptides. So you have one long chain of amino acids right here, one long chain of amino acids right here, and then you have these things called disulfide bonds. All that is when a cysteine sees another cysteine, it's going to create a covalent bond between the sulfurs. So let's review real quick the primary, secondary, tertiary, and quaternary structures of proteins. So remember, your primary structure is going to be the sequence of amino acids, and those amino acids are covalently bonded together. And remember, there's 20 different options, and most proteins are going to be like 400 amino acids long. All right. Then after the amino acids are bonded together, the next thing they're going to do is because of the interactions between amino acids, they're going to form these spirals called alpha helix or alpha helices and pleats called beta sheets. So they're going to spiral and pleat. Then tertiary structure, they're going to start folding in on each other. And so the reason they're going to start folding in on each other has to do, once again, with the interactions between the amino acids. Some amino acids are positively charged, some are negatively charged. So those two are going to be attracted to each other. Some are hydrophilic, some are hydrophobic. So the ones that are hydrophobic are going to bunch up together. The ones that are hydrophilic are going to be kind of on the outside, interacting with the water in the environment. And then quaternary structure is going to be multiple polypeptide chain tertiary structures that come together. And so a protein can have either one tertiary structure, 2, 3, or 4. All right, and so those are the different levels of folding of a protein. Changing even one amino acid from this original chain can completely disrupt the folding process, giving that protein a different shape, and therefore it won't work. All right, so let's go back and let's start and talk about the beginning. So in the nucleus, you have DNA. All right, and the nucleus is able to control the cell by directing the amount and the type of proteins produced by the ribosomes in the cytoplasm and the rough endoplasmic reticulum based on the DNA that's present. So the entire process of DNA to protein is called protein synthesis. And so remember, inside the nucleus, you have chromatin. Chromatin is DNA wrapped around proteins. DNA, what we're going to discuss, the whole overview of this lecture is, how does DNA get transcribed into RNA? How does RNA get translated into protein? All right, so we're going to talk about protein synthesis, and then you have a really nice visual here. Here's the DNA that's inside the nucleus of a eukaryote. It gets transcribed into RNA, still inside the nucleus. RNA then leaves the nucleus and gets translated into your protein, which is your chain of amino acids. So the overview of this lecture is, how exactly is that going to be achieved? So DNA and RNA, let's kind of talk about the differences between them again. They're both nucleic acids, and they're both made up of nucleotides. And remember, a nucleotide is a phosphate, a sugar, and a nitrogenous base. So a phosphate, a sugar, and a nitrogenous base. And so that's the similarity. They're both nucleic acids, and they're both made up of nucleotides. All right. The sugar and phosphate are part of the structure of DNA, but they don't play a direct role in protein synthesis. So the phosphate and the sugar are really a structural component and are not really part of the DNA code. Okay. What really makes up the DNA code are these nitrogenous bases. And so remember, when we talked about replication last week, the DNA code is the T's, the C's, the A's, and the G's, and exactly how those are laid out in the different sequences, okay? Remember, DNA is a double helix. RNA is single-stranded, because DNA is double-stranded, RNA is single-stranded. And then instead of thymine, RNA is going to have uracil. All right, so those are the differences. So I would jot that down, that they're both nucleic acids, they're both made up of nucleotides, and their nucleotides have those 3 parts. However, the nucleotide, the phosphate and the sugar don't really do much, doesn't really code for anything. What codes are the nitrogenous bases. And so in DNA, remember, A pairs with T, C pairs with G. So what you're going to have is, when DNA gets transcribed into RNA, if you see a T on DNA, that is going to be an A on RNA. If you see a C on DNA, that's going to be a G for RNA. If you see an A on DNA, that's going to be a U for RNA. So I'm going to go back over that because that's kind of confusing. I'm going to go through the steps though. So remember, each of the heritable traits in our bodies are the result of our genes or specific segments of DNA. So our traits are coded for by DNA. But is it the DNA itself that is causing your eye color? It's not. So we'll talk about eye color, for example. Eye color is the result of two or more genes that collectively determine the amount of pigment produced by the irises. DNA isn't causing the color directly. It's this protein called melanin that's causing the color directly. So DNA codes for a protein. The protein is then in your eye causing the visual color or the visual trait, right? Or the physical trait. And then this is just showing you, if you see a big letter, that means dominant. If you see a little letter, that means recessive. So the two genes that are responsible for eye color, if they're both dominant, the eyes are going to be darker. And then if they're both recessive, that's when you get little or no melanin produced and you get really light eye color. And so this just kind of goes through all the different combinations of those two genes you can have, dominant or recessive, that causes eye color to become lighter and lighter the more recessive those traits are. And so when we talk about genetics a little bit later this week, I will discuss dominant, recessive, and then how to do Punnett squares for that. All right, so each gene that is transcribed is transcribed for only one polypeptide. And so a protein can be made up of one or more polypeptides. So one protein could be the result of multiple genes, but each gene is going to code for its own chain of amino acids. Polypeptides are produced in the ribosomes, which are in the cytoplasm or they're attached to the ER. Chromatin is too large to exit the nucleus. So a copy of DNA needs to be, a copy of this genetic information needs to be sent from the nucleus out into the cytoplasm. So you can see the dilemma here. Chromatin's too large to exit the nucleus. However, proteins are made outside of the nucleus. So some way the message of DNA needs to get sent out of the nucleus. And it does that through RNA. So DNA gets transcribed into RNA. And then RNA gets translated into protein. And those two processes are called transcription and translation. So DNA to RNA occurs in the nucleus and it's called transcription. RNA gets translated into protein, that's called translation, and that happens in the cytoplasm. All right. So for each gene, DNA is considered a template strand. All right. So let's take this top strand right here. This top strand right here would be the template strand, okay? It then is going to get transcribed into a complementary RNA strand, and this is called mRNA. It's called messenger RNA because it really is the message being sent from the nucleus out to the ribosomes on how to make the protein. So I want you to pay attention to these letters: A, C, C, AAA, CCG, AGT. This is our template strand. Now go down here to see what mRNA was made in response to that template strand, or what mRNA was made to complement that template strand. So A pairs with T in DNA. However, remember, RNA doesn't have T, it has U instead. So if you see an A, that means for RNA you're going to have a U. If you see a C, that means for RNA you're going to have a G. C goes with G. A goes with U, U, U. And then you can see how the complementary RNA strand gets made based on that template. And I think that's going to be the most difficult thing is when I say DNA has an A, you need to tell me that complementary mRNA then has a U. And we're going to do practice on that so that you can kind of get into the habit. All right. Then when RNA leaves the nucleus, it gets read 3 bases at a time. And so I'm going to show you how to read RNA so that you can translate those 3 bases called a codon into an amino acid. So this original DNA template strand codes for this amino acid strand. And then RNA is the messenger between the two. Okay, so let's kind of go back. That was just a big summary of what we're going to talk about. So let's talk about the steps of transcription. So the first thing that happens with transcription, so remember, I want to talk about this. I want to talk about how does DNA get transcribed into RNA. The first step is called initiation. An enzyme called RNA polymerase is going to bind to the promoter of a DNA strand. All right, so initiation. RNA polymerase is an enzyme that binds to the promoter of a DNA strand. That's the first step of transcription. So our goal here is how are we going to get from DNA to RNA? All right. Second step is called elongation. RNA polymerase is going to read the DNA template strand and then is going to add RNA nucleotides that complement it. All right. So which strand is this RNA polymerase actually reading? Is it reading this top strand or is it reading this bottom strand? It's giving complementary bases to the bottom strand. So it's obviously reading the bottom strand, adding complementary bases as it goes. And you can see where there's a T on DNA, it adds an A of RNA. Where there's an A of DNA, it adds a U of RNA. And then C's and G's go together, T goes with A, T goes with A goes with U, and then what would it add right here? It would add a U. All right, so it's making the RNA based on what it's reading from DNA, and it's RNA polymerase that does that. I love transcription because there's only one enzyme. Remember with DNA replication, we had that big long list: helicase, topoisomerase, ligase, DNA polymerase 1, DNA polymerase 2, primase. But what's really cool about transcription is that there's one enzyme. It's RNA polymerase. And this enzyme does everything, okay? The last step of transcription is called termination. So if you follow the pictures down, it's showing you the sequence. RNA polymerase binds to a promoter. RNA polymerase starts reading DNA, puts down RNA that complements the DNA, elongates the strand, it reaches a termination sequence, and it's done. And look at what you're left with is an RNA that was complementary to the DNA template. So once the RNA polymerase reaches like a terminator sequence, you don't even need to know what that sequence is. It's just a certain sequence that signals that, hey, we're done. We transcribed the DNA, we're done. Then RNA polymerase is going to detach. So termination means a terminator sequence was reached. RNA polymerase detaches, releases the fresh transcribed mRNA. mRNA then exits the nucleus and goes to the ribosome. Okay? mRNA is the message that is going to get translated at the ribosome. So the ribosome is the site of protein synthesis. The ribosome doesn't actually do the protein synthesizing itself. What's really doing the synthesizing of the protein is tRNA, all right? Also known as transfer RNA. tRNA reads the message and drops amino acids in as it's reading. So it's like translating, okay? And then the ribosome is just the site where it's occurring. All right. So how does tRNA actually do this? So the mRNA is used to generate a polypeptide at the ribosome. This is called translation. Okay, so I told you what the parts were. mRNA is the message, tRNA is the interpreter, the ribosome is the place. This whole thing is called translation. So we're going from a strand of RNA to an actual polypeptide. So the codons on mRNA are matched up to a complementary anticodon. So this is really confusing. All I want you to know is that tRNA reads mRNA 3 bases at a time and drops off the amino acids that correspond to it. That's all I really want you to know from this slide. Let me repeat that. tRNA is going to read mRNA 3 bases at a time and is going to drop off the the amino acid that matches that RNA codon. Okay? So it carries in the amino acid. There's a new growing polypeptide chain. And I have a video linked on here that I really, really, really need you guys to watch because you need to see an animation of this. Okay. So how does tRNA actually read? And then I'm going to go through some practice on Zoom with this too. This is so difficult to teach not being in person and not having my document camera, but we're going to do the best we can. So the genetic code is called non-ambiguous, meaning that each codon is going to specify one amino acid. Remember, a codon is 3 bases on mRNA. So here's how you read this chart. So let's say mRNA comes in, tRNA is reading it, and tRNA sees AUG. First codon, AUG. So what you do, follow my cursor, you go A, U. So where do A and U meet up? They meet up in this box right here. Then you look to the right and you have G, so it's this bottom one, AUG. And it codes for methionine, shortened as MET, also known as the start codon. All right, and we're going to need to do a lot of practice with this because I'm going to give you codons. You're going to need to tell me what the corresponding amino acid is. So what would CUC be? CUC. You would go to the left, C and U meet up right here. C is going to be LEU, leucine. Not every amino acid chart is going to give you these ones on the side, so I wouldn't rely on that too much. I would just, so C, U, C, they meet up, all 3 of those meet up right here, so it's LEU, also known as leucine. Okay, we're going to do some practice on that in the Zoom, and you do have an assignment today where you're going to actually attempt this. I'm going to show you some more examples here in just a minute, actually, because I don't want to just leave you with that. All right. So AUU, A, U, and then U, ILE, which is called isoleucine. So it's always going to code for that. The genetic code is also redundant. It's redundant, meaning some amino acids have more than one codon. So for example, AUU, AUC, and AUA all code for isoleucine. And so what this does is it kind of covers the cell. It covers it so that if there is a mutation in DNA, so let's say there's a mutation that puts a C here instead of a U, well, they both code for isoleucine, so that would be a silent mutation, so not that big of a deal, right? Because they both code for isoleucine. Whew, thank goodness. We're covered. So redundancy is going to help avoid damage if an error occurs during transcription. All right. This is the amino acid chart right here. Remember, we talked about there being 20 different amino acids, and each amino acid has different characteristics based on the side chain that's hanging off. So these side chains for arginine, histidine, and lysine all have or these 2 right here at least have positive charges. Aspartic acid and glutamic acid have negative charges. So obviously these are going to interact with each other. There's polar uncharged side chains, hydrophobic side chains. So the hydrophobic ones are going to all kind of clump together in the middle. So just depending on what the characteristics are of the side chains hanging off of amino acid is going to dictate how it's going to interact with other amino acids, which is going to lead to the actual folding of the protein. So let's go back to insulin. Let's talk about insulin again. Remember, insulin is a protein hormone that regulates blood sugar. It's made up of 2 polypeptide chains, like I said earlier. And those 2 polypeptide chains are kind of interacting like this. There's some sulfur bonds here. And so let's talk a little bit about how insulin is built and the steps that it goes through. So now I've told you what is transcription, what is translation. Like that's it. That's it. It's not that bad. You do need to go back and review it, but it's really not that bad. It's not as many steps. I don't feel like it's as difficult as DNA replication. Normally though, I would do transcription one day and then I would do translation the next day, but we are kind of pressed for time here, so we're doing them both in one day. So I definitely encourage you to go back, read back through these notes. So let's go through the steps of how insulin is built, and this will help review you on the steps of transcription and translation. So the double helix of DNA is unzipped by helicase. So helicase actually does help unzip DNA so that RNA polymerase can do its thing. All right. The mRNA molecule is then transcribed by RNA polymerase using one of the DNA strands. So this is transcription. RNA gets made. This is inside the nucleus. The RNA molecule that has been freshly made then leaves the nuclear pores and goes to the rough ER where the ribosomes are. Sorry, if you didn't get that down, I'm going to click. The mRNA is then received by a ribosome, which, with the help of tRNA, starts translating the mRNA codons into amino acids. And that's where you would use the codon chart to see what is tRNA going to bring as it's reading the codons. The protein is built one amino acid at a time using tRNA. And then the insulin is sent to the Golgi where it's packaged and it's folded up, it's packaged, and then it's released and exported through the cell membrane. So that's just a broad overview of everything that happens. Okay, so how does a protein actually get folded? The polypeptide chain coils and folds spontaneously, but we have these other protein helpers. So chaperonins are the proteins that kind of help. Chaperonins are kind of like a dressing room. So if you went and you were trying on clothes at a store, you would be like the polypeptide. The chaperonin would be like the dressing room. You would go in, do your thing. And why do we go into dressing rooms? To try on clothes, because we're not just going to do it. Like, we're not going to go into J. Crew and just start taking our clothes off and trying on clothes in the middle of the floor because it's inappropriate and we don't want to cause a scene and we don't want anyone to harass us or do crazy things or anything like that. So we go to a dressing room. It's our protection, right? So a chaperonin would be kind of like the protection for the polypeptide as it folds. Then it leaves the chaperonin. Okay, you definitely need to go and watch this video. I would take a break right now. I would pause, go watch the video so you can get a recap of what happened because this is a difficult topic and we are moving on to meiosis tomorrow. So take a break, go watch this video, then come back and I'm going to finish with talking about genetic mutations and the different types of mutations that can cause misfolded proteins. So I'll see you back in a minute. All right, and so once you have watched that little review video that gives really good animations, I am going to talk about this tomorrow in the Zoom. Yeah, I'm going to talk about this tomorrow in the Zoom. It's really interesting the way that they're making the vaccine for COVID right now. So I'll talk about that because it has everything to do with this chapter, actually. So let's go ahead and talk about mutations and how DNA mutations can cause problems. So any change in the nucleotide sequence of DNA is called a mutation. And so we have different types of mutations. A substitution mutation is going to occur when the wrong nucleotide is inserted for another one. So one nucleotide is substituted for another one. So let's say T is put in instead of A, or G is put in instead of C, something like that. That's going to be a substitution mutation. An insertion mutation is going to occur when an extra nucleotide is inserted. And then you can see with mRNA being translated, it's translated in triplets, right? A codon is 3 letters. So if you insert a letter into DNA, that means that RNA is then going to be misread because it's read 3 at a time. So if you insert one, it's going to shift everything over to the left. And then deletions are going to occur when one nucleotide is deleted. So that causes problems when tRNA is reading mRNA in triplets in the codons. It's going to shift everything over to the right. All right, so I'm going to go back over those different types of mutations and show you some examples. So the first type that I discussed was substitution. Substitution, remember you're substituting one letter for another or one base for another, like an A for a T or a C for a G, and it's an accident. Remember, there's a 1 in 10 billion chance that the wrong nucleotide is going to be laid down by DNA polymerase and it's not going to get noticed or corrected. Remember, I talked about the redundancy of the amino acid code and that some codons will code for the same amino acid. And so let me go back to that. Because that really is important to look at. All right, so if you look at the amino acid code, if there is an accident, there's 4 different accidents that can happen that will end up still coding for leucine. So if the third base here in DNA was anything, A, T, C, or G, still going to code for leucine. All right, and so that's considered redundancy in the code, and that's supposed to kind of cover for any problems. And I'm just going right back to where I was. Sorry to go back. I just wanted y'all to see that. So if there's a DNA mutation that still codes for the same amino acid, it's called a silent mutation. If there is a DNA mutation that results in a different amino acid, it's called a missense mutation. All right. If it results in a premature stop codon, so if you look at your codon chart, some of those codons, those triplets, code for a stop codon, means that the protein is done. It's the end of the protein. That's called a nonsense mutation. All right, so let's look at these different scenarios down here. No mutation, there's DNA, there's RNA. Remember, T, complementary to T is A, complementary to C is G, and then if you looked back at the codon chart, AAG codes for lysine. So that's what's supposed to happen. That's no mutation. All right, let's say C was accidentally substituted for T. All right, so TTT instead of TTC. What the complementary RNA would be AAA, and if you look at the codon chart, AAA still codes for lysine, so that would be considered a silent mutation. All right, let's say DNA, instead of TTC, you got ATC. So the complementary RNA to ATC would be UAG. So if you go and look back at the codon chart, UAG actually codes for stop. So that would be considered a nonsense mutation. Whenever tRNA runs across a codon that's a stop codon, it stops translating and the protein's done. So if you get a premature stop codon, you end up with like this really short polypeptide that just folds into something that definitely is not the protein that it was meant to. And then if you look here, there's conservative and non-conservative. Don't pay attention to that. I just want you to know what a missense mutation is. So the original, remember, was TTC. Let's say this middle one right here was substituted TCC. The complementary RNA is AGG. That codes for arginine. So that's definitely different than the original one. That's considered a missense mutation. And then And the reason why this is called conservative is because it has similar properties to the original one, so it could result in the same folding. Non-conservative would mean it's a way different amino acid. And so this would be missense. It coded for a completely different amino acid, and it's very, very different. It's considered a polar one right here, so that would be a different one that would definitely cause more problems. So some missense mutations are more problematic than others. It just depends on which amino acid is accidentally put in when it's not supposed to. Right? So those are substitution. An example of a substitution mutation that causes a major disease state would be sickle cell anemia. It's the result of a missense mutation in the gene that codes for one of the 4 polypeptides of hemoglobin protein. Hemoglobin protein is a protein in red blood cells that carries oxygen. So if one of those hemoglobin proteins is misshapen, it causes the red blood cells to take on a sickle shape down here. It's like a half moon shape. And what you would have is an inability for it to carry oxygen very well, and then they get stuck in the really small, tiny capillaries. These get stuck and start to build up. So sickle cell anemia can result in blood clots. It can result in fatigue and exhaustion. Really, really bad disease that is only caused by one accidental substitution base. And so what was supposed to be, this is normal, GAG, the accident is GTG. RNA is supposed to be CTC, but the accident causes an RNA to be CAC, and instead of glutamine, you get the amino acid valine, and that totally screws up the folding, which results in a completely misshapen red blood cell. All right, let's talk about insertion and deletion. So insertion and deletions are also called frame shift mutations because they completely disrupt the reading frame. So remember that tRNA is reading the codons 3 at a time. All right, so if you have normal right here, so normal DNA, where's the mutation? It's right here. So look at where my cursor is. This was a substitution. All right, this is a substitution. So substitute one base, it messes up only one base of RNA, messes up one amino acid in the entire strand. Let's say you insert something. So look right here where my cursor is. The C is inserted, so everything gets shifted over to the right. So the reading frame gets shifted over to the right. And tRNA is reading 3 at a time. So instead of reading GAA, now it's reading GGA. But since everything was shifted over to the right, the next one is messed up. Then the next one is messed up. Then the next one is messed up. I hope that makes sense. Maybe I'll probably go over that in the Zoom. So if you insert one here, everything else gets shifted over and the reading frame is shifted. All right. Same thing if you delete one. So let's see, this A up here, it's deleted. Everything gets shifted over to the left because tRNA is reading 3 at a time. It's going to shift everything over to the left, and then all the amino acids are going to be messed up from that point forward. So nearly every amino acid following the mutation will be incorrect, completely altering the structure of a protein. So are substitution mutations serious? Yes, they can be, but a frame shift is always going to be serious. A substitution could be silent, but a frame shift is always going to be serious. So a frame shift mutation is usually going to end in miscarriage. So a lot of times when a woman miscarriages, it's because the offspring had a mutation bad enough to where the pregnancy just couldn't proceed. This is just another example of a genetic disease. I'm not going to read every word on here, but it's called Tay-Sachs disease, and it's from a DNA mutation for a protein enzyme that helps with the breakdown and digestion of lipids. And so and it normally occurs most likely in the Jewish population. And it's considered a point mutation, a substitution mutation, where one amino acid gets changed. And then there's some other things that are going on here, but I just wanted to you guys to see another disease where an abnormal mRNA and an abnormal protein can result in this situation, in lipids not being able to be broken down and digested. And so they get built up in the brain. And what happens is that obviously there's neurological issues. And so one of the first signs is it's like a floppy baby, basically. The baby just has like no muscle tone because of the lipids that have built up in the brain. So it's called Tay-Sachs. All right, last thing is mutagens. So what can cause DNA mutations? The things that can cause DNA mutations are called mutagens. Most of the time, if someone has a genetic disorder, that happened in utero or that happened before conception. Something was wrong with the sperm or the egg to begin with. But sometimes DNA replication can be messed up and mutations can occur after, we've already been made and we're already living and everything like that. And so these would be called mutagens. They can cause new DNA mutations. Ultraviolet radiations like the sun, that's where skin cancer becomes an issue. X-rays, that's why females, when you're getting an X-ray, they'll cover your pelvis with lead so that your eggs don't get mutated. Asbestos, and then tobacco smoke. Tobacco smoke can cause DNA mutations in the cells of the lungs. So definitely smoking or introducing certain chemicals to the body can cause DNA mutations, which can ultimately, these types of mutagens are going to lead to cancer. So what we're going to do next is we are going to, you're going to do some practice just to make sure you understand the process of transcription and translation. And then we're going to cover meiosis, how sperm and egg are made. And then we're going to do genetics, and that will pretty much be the end of the class. All right, I will talk to you guys later.